This protocol provides a long-term in vitro functional method for the evaluation of chimeric antigen receptor or CAR T cells through repetitive tumor cell challenge.
This technique is less time-consuming and less labor-intensive than in vivo CAR T-cell function evaluation, while still achieving an accurate representation of the true anti-tumor efficacy.
This in vitro technique enables the high-throughput screening and differentiation of CAR T-cell antitumor potency, which has the potential application for assessing clinical effectiveness of CAR T-cell products.
Begin by dissociating glioblastoma tumor spheres from a glioblastoma tumor sphere culture with 1 milliliter of cold Accutase and 30 to 60 seconds of pipetting. When the tumor spheres have been disrupted, stop the reaction with 5 milliliters of warm co-culture medium and pellet the tumor cell suspensions by centrifugation. Resuspend the glioblastoma cells to a 1.6 times 10 to the 5th tumor cells per milliliter of fresh co-culture medium concentration, and dilute T cells harvested from a CAR T cell culture to the appropriate percentage of CAR-positive T cells per milliliter of co-culture medium concentration. Next, add 100 microliters of the diluted tumor cells to each well of a 96-well flat-bottom tissue culture plate, and 100 microliters of diluted CAR T cells into each well of tumor cells with gentle mixing. Then place the plate